Good morning everyone. So today we can discuss about culture media classification. So how we can classify different types of culture media. The culture medium or the growth medium, it is a liquid or a gel designed to support the growth of microorganisms. There are different types of media suitable for growing different types of cell. Here we can discuss microbiological cultures which are used for growing microbes such as bacteria or yeast. Culture media contains nutrients and physical growth parameters which are necessary for the microbial growth. All the microorganisms cannot grow in a single culture medium, and in fact many can't grow in any non-culture medium also. So there are certain organisms that cannot grow in the artificial culture medium. They are known as obligate parasites. Examples are Mycobacterium leprae, Rickettsias, Chlamydias and Treponema pallidum. These are obligate parasites. Now coming to the different classification of the culture media based on their consistency. So before that we can discuss the advantages of the solid media as well as liquid media. In the case of solid media, bacteria may be identified by studying the colony character. So we can easily identify the bacteria by studying the shape, size, etc. of the colony. That means the colony characters. And also the mixed bacteria can be separated in the solid media. Solid media is used for the isolation of bacteria as pure culture, and agar is most commonly used to prepare solid media. Agar is polysaccharide extract which is obtained from seaweed, and the agar is an ideal solidifying agent as it is bacteriologically inert, that is no influence on bacterial growth. So the solid medium can be used for the isolation of pure culture. Then this agar, what is the advantages of agar? It is bacteriologically inert and that is no influence on bacterial growth. It remains solid at 37 degrees Celsius, and also it is very transparent also. It remains solid even though at 37 degrees Celsius. Whereas in the case of the liquid media, it is used for the profuse growth. No separate colonies can be visible. Example, the blood culture in liquid media. The liquid media may also consist of the blood culture media. Mixed organisms cannot be separated, such as Streptococcus, etc. So this, if the mixed organisms present in the liquid media we cannot differentiate or culture, for culturing we should inoculate them to the solid media. Now next is the classification of bacterial culture on the basis of its consistency. First of all, solid medium. The solid medium contains agar at a concentration of 1.5 to 2% or some other. Mostly they are inert solidifying agent. Solid media has a physical structure and it may allow the bacteria to grow in physically informative or useful ways. For example, they can be grown as in colonies or in streak line. And the solid medium, it is used for isolating bacteria, and also to identify the colony character of the isolate, we should use solid medium for the growth of the microorganism. Next is semi-solid medium. Semi-solid medium, it is prepared with agar at a concentration of 0.5% less, and it has a soft custard-like consistency and is useful for the cultivation of microaerophilic bacteria or the determination of bacterial motility. So for checking whether the bacteria is motile or not, semi-solid medium can be used. And also it is used for the cultivation of microaerophilic bacteria. Next is about liquid medium. That means broth. This medium contains specific amount of nutrients but not have a trace of gelling agents such as gelatin or agar. Broth medium serves various purposes such as propagation of large number of organisms, fermentation studies and various other trees. For example, sugar fermentation test and MRVP test. Classification of bacterial culture media on the basis of purpose or functional use or application. Many special purpose medium are needed to facilitate recognition, enumeration and isolation of certain types of bacteria. To meet these needs, numerous media are available. So mainly before telling about this medium is nothing but through which the oocysts and what do you mean by this children develop. So the general purpose medium or basic medium is the first example. Basic media are basically simple media that allows most non-fastidious organisms. So fastidious bacteria. So the people want bacteria and nutrient agar because it has. So this is simple medium that supports non-fastidious, means it may grow without any support. Example, peptone water, nutrient broth and nutrient agar. These are considered to be the basal medium. Those individuals are generally used for the primary isolation sending. So these are the examples of the simple or basic medium which are used for the primary isolation. Next is about enriched medium. That means with added growth factors, addition of extra nutrients in the form of blood, serum, egg yolk, etc. to basal medium makes that medium enriched media. Enriched medium are used to grow nutritionally exacting bacteria. The blood, for example for enriched medium is blood agar, chocolate agar, loftless serum slope, etc. Blood agar is prepared by adding 5 to 10% by volume. How much amount of blood? 5 to 10% by volume. Blood is added to the blood agar base. Next is about chocolate agar. It is nothing but when the blood agar is heated, it may causes the lysis of the blood agar and this may result in the formation of chocolate agar. So first picture is nutrient agar. Here the colonies can be separately seen. Second is blood agar. Here we can see isolated colonies and also if hemolysis is present we can see as a zone of inhibition. Next, this is chocolate agar. Chocolate agar, which is a heated blood agar. There are certain organisms which favor the growth of chocolate. That is, certain factors which are responsible for there are certain organisms which grow in chocolate agar are Haemophilus influenzae because this bacteria have nutritional requirements more compared to the normal other types of bacteria. Next is selective and enrichment media. These medium are designed to inhibit unwanted commensal or contaminating bacteria, and so the unwanted contaminating bacteria can be removed in the selective media, and it helps to recover pathogens from the mixture of culture, from the mixture of the bacteria, while selective medium are mainly agar based. It is not liquid based. Enrichment media are liquid inconsistency. So this both selective medium or enrichment medium both have same function: inhibit unwanted organisms, recover pathogens. But selective medium is in solid state and enrichment media is in liquid state. So both this media serve same purpose. Any agar media can be made selective by the addition of certain inhibitory agents that don't affect the pathogen of interest. Various approaches for making the medium selective include what? How to make medium more selective either by the addition of antibiotics or by using certain dyes, chemicals, or altering the pH, or the combination of these three. Okay. The next is the principle of selective medium, that is the differential growth suppression. It is the main principle, which means it is designed to suppress the growth of some microorganisms and while allowing the growth of others. Selective medium is agar based, which means solid medium based, so that individual colonies may be isolated. So this may helps to isolate the exact pathogen from the mixture. Examples of selective media: Thea Martin agar, which is used to recover Neisseria gonorrhoeae, and that medium contains antibiotics vancomycin, colistin, and nystatin. Next is mannitol salt agar. Mannitol salt agar and salt milk agar is used to recover Staphylococcus aureus from 10% NaCl. The potassium tellurate medium used to recover Corynebacterium diphtheria contain 0.04% potassium tellurate. Next example is McConkey's agar. McConkey agar is the most commonly seen agar used for Enterobacteriaceae members that contain bile salt that inhibit the gram-positive bacteria. Next is pseudocell agar, that is also known as cetrimide agar, used to recover Pseudomonas aeruginosa and cetrimide, that is antiseptic agent. Crystal violet used to recover Streptococcus pyogenes containing 0.0002%. Then next is Lowenstein-Jensen medium, which is called LJ medium, used to recover Mycobacterium tuberculosis and is made by incorporating malachite green. So these are all about. So this is LJ medium is used to recover Mycobacterium tuberculosis, and the selective agent added in this medium is malachite green. It is a dye to inhibit the growth of unwanted microorganisms. Only Mycobacterium tuberculosis grows in this media. Next is Wilson and Blair's agar for recovering Salmonella typhi. This is rendered selective by the addition of the dye brilliant green, and this medium is used for recovering Salmonella typhi, and the additional dye add is brilliant green. Next is selective medium such as DCBS agar, used for isolation of Vibrio cholerae from fecal specimens, and they have a high or elevated pH 8.5 to 8.6, which inhibits most other bacteria.